So what causes autism? You know, for decades, this has been one of the biggest questions in science. Today, we're going to be the detectives. We're opening up the case file to try and unravel this mystery one clue at a time. And our investigation, it starts right now. Right off the bat, we hit the classic question, don't we? Nature versus nurture. Is the answer written deep inside our DNA, or is it something that's shaped by the world around us? This is the central conflict that's going to drive our whole investigation.
But before we dive any deeper, we really have to get our heads around this quote from a leading researcher, Daniel Geschwind. The answer isn't a simple this or that. He says it's like pneumonia. There can be lots of different paths that lead to it, so there's no single villain in this story.
Okay, so every good investigation has to start with the basics. Before we can even begin looking for a cause, we have to understand what we're even looking at. We've got to define the autism spectrum enigma itself.
So, autism spectrum disorder, or ASD, isn't just one thing. It's a huge, wide spectrum. It affects about 1 to 2% of kids all around the world, and honestly, how it shows up can be completely different from one person to the next. That word, spectrum, is absolutely key here.
So, when you're trying to figure out the origins of a really complex condition like this, one of the very first places scientists look for clues is within families. You know, does it run in the family? And asking that simple question points us directly toward our first major lead.
And that first lead takes us straight to a major break in the case, the genetic trail. And the evidence we're about to find here is, well, it's powerful, it's compelling, and it pretty much changes everything.
Get ready for this number, 70 to 90%. That is the estimated heritability of autism. To put that in simple terms, it means that the huge majority of the reason one person is autistic and another isn't comes down to their genes. This is the first massive clue in our investigation. And if you're looking for a smoking gun, well, twin studies are basically it. Just look at these concordance rates. If one identical twin, who shares 100% of their DNA, is autistic, there's a more than 90% chance the other one is too. But now look what happens with fraternal twins, who only share about half their DNA. That number just plummets. I mean, the genetic link is pretty undeniable. So this tells us that genes are playing the lead role here, for sure. But, and this is a big but, we are not looking for a single autism gene. That doesn't exist. The genetic map is way more like a vast, intricate web than some simple, straight road.
Okay, so if it's a giant, complicated web, how do we even start to untangle it? Well, the next step in our investigation is to decode the different kinds of genetic clues that scientists have been finding along the way.
The evidence really breaks down into three main categories. You've got the combined effect of lots of common genes. Then there are rare and spontaneous mutations. And finally, there are these bigger, structural changes to our DNA. Let's take a look at each one. First up is what's called polygenetic risk. Think of it like a recipe with thousands of little ingredients. Each one of these common genetic variants is like a tiny pinch of salt or a dash of spice. By itself, its effect is super small. But when you add hundreds or even thousands of them together, they collectively start to shape the final result of how the brain develops.
Next up, we have de novo, which just means new mutations. These are kind of like a sudden spontaneous typo that appears in the genetic code, a typo that wasn't in either of the parents' original copies. They just happen, randomly. And they're really important for explaining cases where there's no family history of autism. They might account for up to a fifth of all cases.
And finally, we get to the biggest changes of all, copy number variations, or CNVs. Now, if de novo mutations are like little typos, think of these as entire paragraphs, or even whole pages, being accidentally deleted or just copied and pasted into the genetic blueprint. They change the dosage of a bunch of genes all at once, and that can have a really significant impact.
So, we have this absolute mountain of genetic evidence. Case closed, right? Well, not quite. Because it turns out, even with a strong genetic predisposition, something else can influence the final outcome. There's another player in this story we haven't talked about yet. And that other player? It's the environment. Now, think of these factors not as the main suspects, but maybe more like accomplices in our mystery, things that can interact with the genetics that are already there. The key thing to notice here, and this is really important, is the timing. All of these environmental risk factors that have been identified happened before or during birth. We're talking about things like the age of the parents, the mother's health during her pregnancy, or maybe exposure to things like heavy air pollution or certain medications while in the womb. These are not direct causes, let's be clear, but they can increase the risk, especially when they're combined with that genetic predisposition.
And this leads us to the biggest, most important question of all. How can something from the outside world, like air pollution or maternal stress, how can it actually reach inside and have a conversation with our DNA? How does the world out there influence the genetic blueprint inside of us?
Well, the answer isn't in one or the other. It's in the interaction between them. This is really the climax of our investigation, where we finally uncover the mechanism that links our two main suspects, our genes and our environment.
And that mechanism is called epigenetics. Here's a way to think about it. If your DNA is the hardware in a computer, epigenetics is the software. It doesn't actually change the letters of your genetic code, but it acts like a whole series of dimmer switches, turning the activity of certain genes up or down based on signals from the environment. It's the bridge that connects the two.
So, what we've really figured out here is that the whole nature versus nurture debate was kind of the wrong question to begin with. It's not a battle. It's a constant, incredibly complex conversation between our genes and our environment, with epigenetics acting as the translator, and all of it shapes how our brains develop.
And once you understand that interaction, it kind of forces you to take a step back and reframe the whole conversation. We start moving away from asking what causes this to a much, much bigger question. How do we understand this?
And this leads to two really different ways of thinking. On one hand, you have the traditional pathology paradigm, which has always looked at autism through a medical lens, as a disorder that's defined by a list of deficits that need to be, cured or fixed. But there's a different perspective that's really gaining ground, called the neurodiversity paradigm. And it reframes autism not as a broken version of normal, but as a natural and valid form of human variation, one that comes with its own unique set of strengths, its own perspectives, and yes, its own challenges.
And this brings us right here to our final thought-provoking question. As we unravel all of this beautiful complexity, we have to ask ourselves, is the ultimate goal of all this investigation to pinpoint one single cause so we can prevent or cure it? Or is the goal to better understand, accommodate, and support the full, diverse spectrum of the human mind? The answer to that question is going to shape the future of research, and way more importantly, it's going to shape the lives of millions.